Hi everyone. So I did have a recording, actually I had two recordings of my proposal seminar. And what ended up happening was some of the audio cut out on one and some of the video cut out on the other. And instead of trying to edit and mash them together, I figured it would probably just be easier for me to go through my proposal again, and just do this. So the title of my proposal is Impact of Selective Preferences of Vertebrate Scavenging on Arthropod Communities Associated with Carrion of Varying Biomass and Number Across Seasons. So to start, I would like to ask you a question, or a rhetorical question. Picture a rainforest, and in that rainforest, 58% of the biomass is dead. What does it look like? Does it look something like this, where the rainforest had either been deforested, or maybe there was a forest fire, or perhaps some natural disaster? What if I told you it looked a lot like this, where an Odom 1970 found that 58% of the life in a Puerto Rican rainforest was made up of dead plants and animals. And of that 58%, carrion was 1% of that. And that doesn't sound like a whole lot. But when you think of it on a global scale, that's about 10% of the world's biomass is made up of dead animals, or carrion. And carrion is called an ephemeral resource. This means that it is spatially and temporally stochastic and short-lived. In addition, this resource is extremely nutrient dense, making it the site for intense competition, particularly between microbes, vertebrate and invertebrate animals. And the animals will consume these tissues, and the microbes will make the nutrients available for plants. This process, because it's from intense competition, will happen pretty quickly and happen basically out of our view. But what happens if there's so many animals that die at once that it's really hard to ignore? This is a photo taken from Australia when over 5,000 bats and flying foxes had died due to a heat stroke, basically. So the temperature had reached 43 degrees Celsius, which is about 111 degrees Fahrenheit, killing all of these bats and flying foxes. And these events are called mass mortality events, which has formally been defined as a significant amount of population lost within a given year. The number is approximately 25%, though these numbers can change drastically depending on the populations that you're looking at. So a couple of examples that you may be aware of is 300 reindeer dying in Norway due to a lightning strike, as well as 200,000 saiga antelope in Kazakhstan dying due to disease. And unfortunately, these mortality events are increasing across taxa in magnitude and frequency, leading us to two major questions. What are the causes of these events, which is oftentimes determined shortly after the event has occurred? And what are the ecological impacts of these events? Specifically, how does the ecosystem respond to these events? And how does biomass play into this? Mass mortality events can be considered a disturbance, which is the loss or destruction of biomass. And when you're in my lab, you know that biomass is also a dead thing. So I'm going to edit this to say living biomass. And if you look at this graph by Zach 2016, this is looking at, on the upper graph, primary producers, and on this lower graph, elemental outputs. So after a destructive event or a disturbance, primary producers will die off, reducing net productivity, and the elemental outputs will increase significantly from these plants and animals decomposing. And then over time, the nutrients will disperse throughout the environment, and these primary producers will increase in abundance, having a bottom-up effect on the environment. However, not all disturbances are bad. Some disturbances can actually increase the biodiversity and function in an ecosystem. So in this example, we're looking at butterfly diversity and richness. And during controlled forest burns, there was an increase in richness and diversity. And on top of this, this can change per ecosystem. So when you look at a forest habitat versus a riparian habitat, the riparian habitat had a higher richness and diversity in comparison to the forest. And in addition, the diversity that was present before a disturbance event can also mediate the severity of that disturbance. So this is because of something called functional redundancy, where in this example you have two predators. If you lose one of the species in a disturbance event, the other predator is still maintaining that function. So in disturbance events, the ecosystem can respond largely in one of two ways. Either it is resistant or it's resilient. And when we're talking about resistance, it's when the ecosystem maintains the same approximate function after the disturbance has occurred. When the ecosystem is resilient, after the disturbance, the function is lost, but over time, the approximate function is returned. And as Periclesis had said, dose does make the poison. So depending on the severity and frequency of this disturbance, ecosystems can respond differently. So a previously resistant community can then become less resistant and losing that function. And this can be assisted in the intermediate disturbance hypothesis, where low disturbances in intensity or frequency and high disturbances can have a reduced species diversity. So if we look at low disturbance events, the species diversity is lower because individuals can competitively exclude these resources. And when the disturbance is extremely high, you have extremely tolerant species colonizing areas and monopolizing that area. But when you have intermediate disturbances, there is enough disturbance in the area to knock down very competitive species, but not enough to completely wipe out other populations. And when we look in relation to carrion biomass, we know from the studies that we have in literature that lower biomasses have less species diversity and richness than higher biomasses. And these studies look often at rats, rabbits, and things as large as pigs or dogs. And we also know that until recent years, these animals were placed close enough that we can consider them mini mortalities, where the biomass was actually accumulating and these weren't single pigs. So these masses can be anywhere from 2 pigs to 5 pigs. And these studies have been pretty useful. We know that through these studies, insects play a major role in the rate of decomposition. So on your right, you see that insect access, and on your left, you see no insect access. And with insect access, carcasses were completely consumed, where with no insect access, they mummified. And this causes abiotic factors to be the mechanism in which these carcasses will decompose. And when we study the arthropod community, we typically cage our carcasses. And when we do this, we're excluding a large array of biodiversity that feeds directly on the carcass and on the individuals feeding on the carcass. And in addition to this, we also know that vertebrate scavengers, in addition to arthropods, accelerate the rate of decomposition. So if you see on your left, you see scavenger and insect access alone. And this was seen across biomass. So when we look back at the intermediate disturbance hypothesis in relation to carrion biomass, we see that we know a lot about the lower end of the curve, but we don't know too much about the higher end. So the highest being these mass mortality events. And we also don't know the trajectory in relation to vertebrate scavengers if they mitigate the effect of high biomasses, lessening this impact on lower diversity in these high disturbance areas. And what we need to know is the in-between these mass mortality events and high biomass. So what I would like to propose is my objectives for a mass mortality event, where we would look at how the ecosystem responds to mass mortality, how biomass plays an effect on this, and determine if vertebrate scavengers have another effect on these. To do this, we have 5 sites ranging in biomass between 25 kilograms and 725 kilograms, with each site being distanced by 1 kilometer. At each site, there are 2 biomasses two equal biomasses, where one is fenced and the other one is open, with 100 meters between each of these masses. The fencing is a 5 by 10 centimeter fencing with bird netting over the top to prevent scavengers. And at all of our sites and plots, we had 4 sticky traps facing the inside of the biomass, with them each being 30 centimeters from the ground. All the sticky traps were placed 2 weeks prior to the pig's placement, and then throughout the study either as the traps filled or after a 2-week period. What we'll be doing is a mixed effect model looking at time, biomass, and fencing to answer my objective questions, or my objectives rather. And we would look at the following community metrics. So functional richness or feeding guild, so this could be anything from predators to detritivores, what they're playing as their ecological role, as well as family richness, diversity, and abundance of these arthropods. So what I'm going to show you is a series of different statistics that I'm not proposing, that I've done since my time starting here. So this first one is called a cluster analysis, and you can think of it similar to a phylogenetic tree, where the branches that are closest together or most similar, and the ones furthest away from each other are most dissimilar. So when we look at the placement of the before pig placement, we see that all these are clustered together, and directly after pig placement, these are the most dissimilar sites. And over time, it starts to approach back to what the sites looked like before the pigs were placed. This suggests that, at least with the preliminary data, that the ecosystem is showing resilience. When we look at dipteran diversity, we can see that across sites, before pigs were placed, there was no significant difference. This is through a test called rarefaction curves that allow for a small sample size, since this is a preliminary data set. And when we see post-pig, that there is a significant difference, and at the 2 extreme biomasses, so the smallest and the largest biomass, we see the lowest amount of diversity, and our moderate biomass has the highest diversity. And this will follow, or this does follow, the intermediate disturbance hypothesis. When we look at vertebrate scavengers, we see that our moderate biomass, 181 kilograms, actually had no impact between open and fenced sites. But when we look at our higher biomass, 363 kilograms, we see that the open sites allow for a higher abundance of arthropods than our fenced site, which is pretty interesting. And when we looked at what vertebrate scavengers were present, we saw that vultures were actually the most abundant scavenger, which isn't exactly surprising. But it is important because of the vulture feeding biology. So vultures will start feeding from the orifices and move their way to the thoracic cavity. So they'll start feeding on soft tissues, and they'll do this in a jabbing and tearing motion. And when they're doing this, they're creating open wounds, and these wounds are readily available for primary colonizers to oviposit in, which they do in forensics cases, post-mortem, and in the natural openings. So with an increase in openings, either from number of carcasses or vertebrate scavengers creating these openings, we have an increase in surface area to volume ratio, and in turn, we have an increase in insects colonizing the carcasses. So we can project that as carrion biomasses increase with vertebrate scavenger presence, the diversity is mitigated by high carrion biomass, and we see that is standing out. But this is only projected to what we know. In the dotted line, we do not know in these extremely high biomasses what the diversity would look like. So while we've been focusing on this high end of the graph, I do want to draw your attention to the lower end of the graph, because this is equally as important as high biomass. So when we have small carcasses, it is extremely prone to competitive exclusion. And this is a really hot topic in our lab and other labs in the department. So Constance Lynn in Dr. Micky Eubanks' lab is looking at fire ants in competition with blowflies. Jenny in our lab is looking at necrophorus beetles in competition with blowflies. Casey in our lab is looking at microbes in competition with blowflies, and if you couldn't guess, I wanted to look at vertebrate scavengers in competition with our blowflies. And the reason why I chose dipterans in competition with vertebrates is because they encompass most of the arthropod community. And the families that we most likely see at carcasses are caliphorids or our blowflies, sarcophagids or our flesh flies, and muscids or our houseflies, with caliphorids being the most abundant. And this is because they're necrophagous as larvae, meaning that from the time that they're laid on a carcass as an egg, they will utilize the carcass, feed on it until they're third instars. And then they'll end up leaving the carcass, pupate in the environment, and become an adult. And this dependency on the carcass is important in competition with vertebrate scavengers because these flies need the carcass to survive. So remember earlier I had mentioned that carcasses are ephemeral, they're nutrient dense, and they're the site for intense competition. And we talked about how biomass does have an effect on the intensity of this competition, but there are also abiotic factors that are involved. So in relation to vertebrate scavengers, feeding relies on the likelihood that they'll locate a carcass and the risk assessment associated with it. Particularly, is the habitat safe? And is the risk of consuming this carcass lower than the reward of consuming it? So there's several different factors that are influencing vertebrate scavenging. So for habitat, open habitats allow for site-reliant scavenging. However, it makes them more prone to predation, where wooded habitats have more areas to hide, if you will. In season, higher humidity and temperature favor insect and microbial development. And we see this in vertebrate scavenger studies, where in 20 degrees Celsius or above, so in summer, we see 20% scavenging, and in the winter, or 18 degrees Celsius or less, we have up to 50% scavenging. And there has been a lot of studies done on carcass types for scavenger ecologists, on species and other metrics, but there hasn't been anything done on fur colors. So we would assume that high contrast fur colors would also allow for site-reliant scavenging. So the objectives in my study is to determine what habitat, season, and carcass type provides the greatest competitive success for our blowflies. So carcass types being broiler chicks, brown rats, or white rats. And to do this, we had two fields, Coulter Airfield in Bryan, Texas, and the Texas A&M Farm in Snook, where we had an open site and wooded habitat. And this is what the habitats looked like on the ground, where the open habitats had low grass and no tree cover, and the wooded habitats had tree cover and some shrubbery. At each of the fields, there were 12 carcasses per habitat, 4 of each carcass type, each being greater than 50 meters from the next. There were 3 game cameras, one at each carcass type, and 1 data logger placed in one of the cameras. Accumulated degree days is a way to take temperature and time and relate them to different things. So we use this in forensics to basically backtrack and figure out when flies more likely colonized a carcass. So we can do the inverse of this. So if we want to look at when we place carcasses down, if flies were to colonize them immediately, when would they be feeding third instars to conclude the study? So we would want to pick up these carcasses right before the larvae reach this pre-pupal stage and leave the carcass and are free living. So we did this twice over the summer, once in July and once in August in 2018 and 2019. And we calculated this out to be 3 days based on Cochleomyia macellaria. And in the winter, we've done this once in December, and we'll do it again in 2019. And we used the Formia Regina data set, and we calculated this out to be 63 days. Though after just 8 days, all of the carcasses were consumed by our vertebrate scavengers. So this concluded the study. And For the duration of the study, we checked the carcasses at dawn and dusk for scavenging and insect activity. And at the end of the study, which was at the end of this development time, we took the carcasses with maggots back to the lab and reared them out, and then identified species, or identified flies down to species, rather. So with this, we did relative risk and chi-square for vertebrate scavenging and the utilization by primary colonizers. And we're going to be doing an ANOVA and a Tukey's HSD for abundance, species richness, and Shannon diversity of these flies. So even though we were looking at vertebrate scavengers, we know that fire ants did play an important role. So if you see here, this blowfly is trying to oviposit onto this carcass. The fire ants are attacking the fly, making them fly away. So when we look at red imported fire ants and vertebrate scavengers completely monopolizing these carcasses, vertebrate scavengers were more likely to monopolize these carcasses than our fire ants. And when we look at open and wooded habitats, white rats and chicks were more likely to colonize in wooded habitats than any other carcass. And in both open and wooded habitats, chicks were more likely to be colonized by or consumed by red imported fire ants. When we look at the primary colonizers and what's left, we can see that, of course, winter does not have any utilization by primary colonizers because they were consumed completely by vertebrate scavengers. So when we focus on summer, generally speaking, there is no preference in open or wooded habitats across carcasses. But when we look at carcass specifics, we see that rats were more likely to be colonized than chicks, and brown rats were more likely to be colonized than any other carcass in wooded habitats. And when we look at our vertebrate scavengers, we can see that we did have armadillos, but they didn't consume our carcasses. They were consuming either the insects out of the carcasses or they were just playing with the carcasses. We just saw them tossing them around a little bit and then they would leave. Vertebrate scavengers such as vultures would be highly present at our carcasses, as well as mesopredators such as foxes, possums, skunks, and coyotes. When we look at what is the most abundant primary colonizer, it was Lucilia exemia, which isn't shocking because it is a known carrion colonizer, though it is relatively rare in the U.S., being limited to Florida and Texas. What we would have expected to see was Cochleomya macellaria and Chrysomya rufifasces, as these are the most abundant flies in the area in the summer. Though we did see Chrysomya rufifasces with Lucilia exemia, which isn't surprising because they have been observed to cohabitate in Texas. So when we look at vertebrate scavenging, if you recall, I had said in the summer, previous studies have seen that up to 20% scavenging and in the winter up to 50%. But in this study, we've seen anywhere from 53 to 91% scavenging in the summer and 100% scavenging in the winter. So the next question is why is scavenging so high and compared to what we've expected to see? And that could be because of anthropogenic land use, which disrupts the ecology and population dynamics of wildlife in the area. So we know that fragmented landscapes negate temperature effects in relation to scavenging, where up to 96% can be scavenged despite these high temperatures. And this can be because of trophic downgrading. So when you lose these apex predators, there is a mesopredator release, so they're more abundant in the area, making it more likely that they're going to find these carcasses. So what does this mean to dipterans? And I would like to generate a scenario where you have a forest habitat with multiple different carcasses, and vertebrate scavengers consume these carcasses, taking them from the environment. This allows for flies to only colonize what's left, and even after these flies have colonized these carcasses, the ones that vertebrate scavengers are selecting for can still be removed from the environment. So after several generations of these flies colonizing these single carcasses, if one of the previous carcass type that was selected for by vertebrate scavengers is left, will flies still colonize it? And is the selection even possible? So we do know that there are selection preferences in flies across season, time of decomposition, and carcass size. For example, Cochleomine macellaria actually prefers larger carcasses. And this is due to something called niche partitioning, where either a resource or a space and time is utilized as a resource. And you have something called the fundamental and the realized niche. So the fundamental niche is the entirety of your niche. So if we want to talk about areas you can colonize on a carcass, blowflies can colonize an entirety of a carcass if it's just themselves. But if you're in the face of a competitor, you have to share this niche, so you'll have a subset of this. So in the face of a competitor, these blowflies are located to a smaller portion of their resource. So these are the questions that I have proposed in my written proposal, though I still had a couple unanswered questions that were just itching at me. So one of them being, is the scavenging that we see in these environments truly color driven, or are there some type of olfactory cues that are involved with these coat colors? And as the scenario I proposed earlier is potential, could we actually select for these types of carcasses in blowflies and have them colonize these carcasses? So volatile organic compounds, or VOCs, are airborne molecules that allow you to detect through smell. We know that carcasses produce significantly different volatile profiles across species. We also know that within species there are variations in volatile profiles depending on feeding types and diseases as well as other things. But can coat color have a significant difference? So to do this, we would do traditional volatile pulls, that the methods that I've gotten from Xanthecose, which is, and modified them a little bit, of course. So we had 6 chicks, white rats, and brown rats. And we will be pulling at 2 hours, 24 hours, and 48 hours. And to do this, we'll put them in these little tins and put aluminum foil over them and let them accumulate for 1 hour. And then we'll take the tins to our machine and we will extract these volatiles for an hour at 1 liter per minute. So we'll then elute these compounds using dichloromethane and make them into a liquid, and then we'll add liquid octane to them and bring them to our GCMS, where they will tell us what compounds are present. So then we'll go ahead and we'll identify and quantify the major compounds in these different carcasses, and we'll use something called a PCA, which can differentiate the different compounds and tell us what is significantly different, as well as a species indicator analysis, which will tell us what's driving these differences. So we know that volatiles are used for our carrion flies to locate and select for carcasses. And we also know that we can train insects to go towards volatiles that are not necessarily advantageous for them. So in this example of the olive fruit fly, these flies were trained to go to volatiles that were previously repulsive. By when they did go to this resource, they were given a food source. And this was done over and over again until they were selecting these volatiles even though there was no food present. And this is important for oviposition because of the mother's knows best. So animals will look for a resource to lay or raise their offspring on that they believe is the most suitable for their offspring's success. Emerald ash borers are known to do this with ash trees, as they will colonize the same carcass or the same trees as they had when they were offspring and raise their offspring on them. We don't know if this is from associative memory, similar to what the olive fruit fly has done, or from chemical legacy, which is the larvae will consume these resources, and in their pupae, they're surrounded by these chemicals as they are becoming adults. And then they will have a genetic change that will allow for them to select for these volatile cues. So the question is, can Luciliexemia, regardless of how this happens, exhibit an oviposition substrate preference. And to do this, we have two colonies. So we have a beef colony and a chick colony, where we're up to the second generation now, but we want to raise them at least to 10 generations to create bottlenecking so we can have these selective lines on these specific carcasses. And then each of the colony will be subjected to a series of choice tests. So at age of 14, they'll be provided a standard protein source to make them gravid. And then we'll put 200 flies, male and female, in these cubes. And these cubes were tested and the methods refined through Casey Flint, where she had these flies, put sticky traps on the arms, and at the edge of these had mesh to prevent flies from colonizing whatever substrate you had on the ends, and a light on top. And this was generated over a 2-hour period to allow for flies to select. They would land on the sticky trap. You can pull the sticky trap out and quantify how many flies have selected for that side, or have landed in that side, rather. So we'll have different choices. So the first one will be for chicks against a control, which is nothing, beef liver against a control, and chicks against beef liver. And this is what this looks like in real life. This is a picture from Casey's study with her rats against a control. And she has a sheet to prevent light from being a factor in these choices. So we'll have 6 cubes per treatment, as I had described, and we'll do this across 3 different days to see if there's a time effect. And then we'll repeat this whole study 3 different times. So after talking about mass mortality events and bringing it all the way out to selection of carcasses, what does this mean in entirety? So mass mortalities don't impact one species in general. They can impact a large array of species, and these species can be distributed in different distances from each other. So what I wanted to do is propose a new model based on the intermediate disturbance in relation to vertebrate scavengers. So along the x-axis, we have carrion biomass, just as we had with the intermediate disturbance, where we have a low disturbance and a high disturbance. And then we have an arthropod community metric. This can be anything from richness, diversity, to abundance. And when we have one carcass, so we can have low biomasses being a single rat versus high biomasses being a single whale. And this is what we would project to see if there was no vertebrate scavengers involved. And if we had 100 carcasses, we'd have more surface area. And we can have either 100 rats versus 100 elk versus 100 deer. And we can keep increasing the number of carcasses until we reach this critical mass, where the maximum arthropods available in the environment, regardless of the number of carcasses, are met. So more arthropods are not able to colonize these carcasses just because they do not exist. And hopefully by now you understand that vertebrates are really important to arthropod communities and their dynamics on carcasses. So we wanted to take into account vertebrate satiation, which is basically the vertebrate community as a whole being full, if you will. And depending on the ecosystem, this line can change, so it's not static. So in some ecosystems, vertebrate satiation can happen at lower biomasses or higher biomasses than what I'm going to talk about. And depending on the number of carcasses and the biomass, this satiation can swing like a pendulum. So what I wanted to do is have this null standard of what we would predict to see. So with one carcass, you would project that in low biomasses, there's competitive exclusion going on. And as we reach vertebrate satiation, arthropods are allowed to consume some of these carcasses as well, and some of this biomass. And as we pass vertebrate satiation, the effective biomass is mitigated. And we would expect to see this just like when vertebrate scavengers are not present, to increase until we reach this maximum threshold. Though this is where we've finished our research so far, we project that this could continue on until there's some biomass where vertebrate scavengers no longer mitigate these effects of biomass. So when we put these two together, this is what this looks like. So the question is why does biomass and number of carcasses and presence of vertebrate scavengers, why are they important? Well, when we look in the eyes of feeding dipterans, overabundant dipterans are not necessarily the best for the environment. We know that blowflies produce a parasitic behavior called myiasis, where they'll colonize living individuals, consuming necrotic tissue and sometimes living tissue. This can increase the rate at which animals decompose in an area that have died near biomass mortality event. This can not only release nutrients into the environment faster, but it can also affect TOC estimates when in relation to forensic science. In addition, these flies are known to spread pathogens. So in a study by Pace et al., they found that livestock presence and feces of livestock where these flies were colonizing were spreading pathogens from this feces, such as E. coli and salmonella, and spreading it onto crops. And when we look at animals, they have been known to do this with anthrax as well. So in this case, there was exotic goats that were, one individual had died from anthrax. These flies had colonized the carcass, and then they had spread the pathogen to the other exotics on the farm, killing them as well. Which is alarming because diseases are the major cause of mass mortality events in recent years. So I would like to have another scenario based on the model I proposed. So if you have a single mass mortality event where the threshold of maximum arthropods are met, and these flies can lay up to 200 eggs, and they can do this up to 7 times. So they're growing at an increasing rate in abundance over time. This maximum arthropod threshold can also lift, and in subsequent mass mortality events, if this is from disease, can cause another one to occur where this maximum threshold can be reached again, and this can happen over and over and over again, but we don't know where either this stops, the effects on the ecosystem, or how it can even be prevented. So I just want to reiterate on these graphs what we know and what we don't know. So we know up to a point that vertebrate scavengers can mediate the effect of high disturbances in relation to carrion biomass in the arthropod community. But we don't know when this drops off. And the question is, can we ever know? Can we ever generate enough biomass in a study to look at this? And we might not have to. So there are mass mortality events happening around the world, and if we studied them as they occurred, looking at the arthropod community, then we can better understand them. And while we think that these events are pretty unpredictable, they might not be as unpredictable as we originally thought. So there are several examples where these events can either be predicted because of a high influx of individuals into an area, such as in Alaska with salmon runs. This mass mortality actually occurred from increasing temperatures in the water. And in India, where birds have been known every year after the monsoon season to virtually commit suicide every day at a particular time. So when we see events like these occurring around the world that we may not always know the answer as to why they're happening, we can still use these as a way to study the effects ecologically of these events. So with that, I wanted to acknowledge the slew of people who've made this possible. Anywhere from research, so everyone at Mississippi State who set up the mass mortality and collected the sticky traps throughout the study. I had no part in that. I can't take any recognition for that. That was all done by the hardworking students at Mississippi State. Amy and Abby for helping me identify individuals on sticky traps down to species, or down to family rather, as well as David, Lauren, and Sophia for going out into the field with me on my scavenger project, Dr. James Beasley for allowing us to use his game cameras, And of course, my lab family and my family for helping me watch Emery. Amy also took a huge part in watching Emery in the first 6 months of her life, as well as Laura Tomberlin, allowing me to go to classes and get things done with a newborn, which was a whole lot of help. And especially my husband, who takes care of my daughter and does a lot more for me when I need to get things done for research than I think I give him credit for on most days. So without that and without all of these people, none of this would have been possible. And with that, I'll take any questions.